So I'm going to be presenting on my current research in hemp biology. My name is Janae Heiss. So this is going to be the way the presentation is structured. First, we're going to have the introduction, and then we're going to go over the objectives of the project. I'm going to talk about the different project overview stuff, so the different sides of background that we have for this project, and then we're going to get into the methods, results, and then questions. So for an introduction, this is my hemp research project. It started in August 2019. The hemp samples were collected from September to November in 2019 and 2020. And samples were collected from local hemp growers in Maryland. Because cannabis sativa is legal, it's important to try to figure out these important things to continue making it a better agricultural crop. So we need to perform more research into hemp in general, learn what conditions are ideal for hemp in Maryland, determine the genetic variation that's present in existing hemp strains, and determine practical chemical content for existing hemp strains. So as a grad student at Frostburg State University, I am going to do a study on this. I'm doing a study on this. The objectives of this study, our question is, what is the genetic diversity and chemical profile for hemp in Maryland? We want to determine hemp's requirements for high CBD production and genetic diversity in Maryland. The research will examine the genetic diversity, chemical composition, and the correlation between the two. So hemp or marijuana? Hemp's scientific name is Cannabis sativa L. Hemp and marijuana are determined based on the total tetrahydrocannabinol or THC concentration. So the total THC concentration is based on delta 9 THC and delta 9 THCA. If there is over 0.3%, it's marijuana. If it's under 0.3%, it's hemp. The negligence threshold for that is currently at 1%. So getting into legal information. Until 1937, hemp was commonly grown in the U.S., but then the Marijuana Tax Act of 1937 happened. It was done in response to concerns about increasing marijuana smokers, and it put hefty fines on the growing of any sort of hemp or marijuana. And then in 1970, the Controlled Substance Act was enacted, which made cannabis sativa a Schedule I drug, so no one could grow it anymore. In 2014, though, the Federal Farm Bill was enacted, which legalized hemp for research purposes again. In 2018, the Hemp Farming Act of 2018 nationally legalized hemp for research and cultivation. So it took hemp off of that controlled substance list. So now it can be legally grown. In 2019, the USDA did an interim ruling which provided more freedoms for transporting, selling, and distributing hemp products, but it did increase THC restrictions and limited testing times. So in 2015 of 2021, there was a new update. So this year, we get a final rule for the Hemp Farming Act of 2018, and it is effective on March 22nd. It builds on the interim final rule, with changes including the negligence violation was raised from 0.5 to 1%, non-compliant plant disposal methods were expanded, non-DEA labs can test hemp until next year, and then sample collection was changed. So samples can now be collected 30 days before harvest. They can be taken from more of the plant and from fewer plants, so that's better for the growers. General biology, next part of hemp. For the general biology of hemp, it is a wind-pollinated plant, it grows rapidly, and it's currently being considered for phytoremediation. It's grown from seeds or cuttings, which are clones, and it's grown for three main crops: fiber, seed, and flower. It takes hemp 6 to 8 weeks to grow to maturity in a greenhouse, and 4 to 11 weeks to flower after the flower induction signal. What exactly that signal is that makes hemp flower is debated. So it's commonly considered to be light dependent, so it's a short day plant maybe, They think 12 hours light, 12 hours dark for floral development, but newer research has shown that it's day neutral for flower induction. So light doesn't affect the flower induction, but it's short day to make the flowers mature. Hemp flowers. Hemp is a dioecious annual with imperfect flowers. So dioecious means it has two growth forms, and annual means it only lives for 1 year. The male flowers have tepals, stamens, and pedicels, so they have only whorl 3 and a combined whorl 1 and 2. And then the female flowers are made of subsessile pairs. They have an ovary, a stigma, and a style. They only have whorl 4 and a combined whorl 1 and 2. So what are these whorls I'm talking about? Basically, there is floral development, and overall flowers are divided into 4 whorls. A complete whorl or a complete flower has a whorl of sepals, a whorl of petals, a whorl of stamens, and a whorl of carpels. Because hemp has male and female growth forms, the female form does not have whorl 3, which is distinctly male, and the male form does not have whorl 4, which is distinctly female. Oh, this is a picture of 2 cannabis plants growing beside each other. This one is a male, this one is a female. So the one on the left is the male, the one on the right is the female. Female flowers, they have inflorescences, which are clusters of flowers arranged on a joined stem. They are commonly called colas. They are made of, the plant has trichomes, which are small hair-like structures that help protect the plant from herbivory. The trichomes are specifically glandular, which means that they have secretory glands. And those glandular trichomes are present all over the leaves and flowers. Those trichomes are specialized to produce and accumulate terpenes specifically, and they produce the cannabinoids. So when you want chemical content, you want those glandular trichomes. So cannabinoids. More than 180 cannabinoids have been discovered. All of them have 22 carbon molecules in their acid form and 21 molecules in their decarboxylated form. Cannabinoids are terpenophenolics, which means that they're synthesized from terpenes and phenols. GPP, which is a terpene precursor, reacts with oliptolic acid, a phenol, to make cannabigyrolic acid, CBGA. CBGA then makes all the other cannabinoids. The main cannabinoids in hemp are THC, CBD, CBG, CBC, and CBN, and each of them has an acid form more commonly found in live plants. So THC and THCA, THC is psychoactive and it has a higher risk of adverse effects. The acid form is most commonly found in live plants. So the difference between the two is listed here. If you'll notice on the one side of the molecule, it has a carboxyl group with an extra 2 oxygen molecules on the bottom one that it doesn't have on the top one. So that's the difference between THC and THCA. The total THC is used to determine drug versus medicinal hemp. Delta 9 THC is used for the total THC, and it has a double bond between the 9th and 10th carbon. Delta 8 THC exists, but it's much less common than the Delta 9 THC. Delta 8 THC has a double bond between the 8th and 9th carbon. There's also CBD and CBDA. These are the most medicinally important compounds. They offset the symptoms of pure THC, and they're used to treat neurological disorders, fracture healing, diabetes, epilepsy, cancer, and they can enhance bone healing by aiding in collagen cross-linking. They are antibacterial, antioxidant, and anti-inflammatory. And CBG, CBGA, and CBDA are sometimes included on the total CBD content of products. Then there's the rest of the cannabinoid molecules. So CBG, CBC, and CBN. CBG and CBGA are the precursors to all the cannabinoids. They're antimicrobial and anti-inflammatory. CBC and CBCA are more prevalent in young plants. They have sedative, anti-inflammatory, antibacterial, and antifungal effects. And lastly, there's CBN and CBNA. They're found in aged cannabis, and they're a degradation product of THC. Next, we have the terpenes. Terpenes are a group of molecules that are insoluble in water. More than 100 of them have been found in hemp, and they influence the medicinal properties of hemp. Any sort of aromatic plant generally has a terpene associated with that. So things like mint are terpenes. They act synergistically to enhance different properties. So they can be primary or secondary metabolites. Primary metabolites means that they're needed for the plant to live. So without this primary metabolite, the plant would just die. And then secondary metabolites are needed for the plant to thrive in the ecosystem. So some of these terpenes are made for protection against insects. So just because they're not there doesn't mean the plant wouldn't live, but it wouldn't be able to do as well as plants that do have those secondary metabolites. There are two different types of terpenes in hemp. There are monoterpenes, which are 10 carbon compounds, and they include beta-myrcene, pinene, terpinolene, oxamine, and limonene. Out of those, beta-myrcene is the most common. It has anti-inflammatory and anxiolytic effects. There's also sequesterpenes, and they're 15 carbon compounds. And out of them, beta-carotene is the most common. It has anti-proliferative effects. So now we're going to talk about hemp genetics. Hemp DNA, it has a labile sex system. So hemp sex changes in response to the environment and stress. It has 10 total chromosomes. There are 9 diploid autosomes, and then an XX or an XY chromosome. Hemp's genome has been sequenced, and there's been microsatellite work done. So in the early 2000s, the first microsatellites were tested, and then in 2012, the first STR multiplex was developed. That was used in police investigations into determining whether things were drug type or just field hemp. There was also a 2017 verification study of microsatellites done to make sure that all of those microsatellites worked and were well developed. So, genetic techniques. You've heard me mention microsatellite so far in this presentation, but you might be wondering, what is a microsatellite? A microsatellite is a segment of repeating DNA that's between 1 and 6 nucleotides. It's known as an STR or an SSR. STR stands for short tandem repeat, and SSR stands for simple sequence repeat. All three of these terms refer to the same exact thing, but they're just in different fields of biology. Microsatellite is more general, whereas forensics uses STRs and botany uses SSRs. These microsatellites are found by scanning the genome for repeats and filtering to determine the potential for self-complementarity and off-target binding. And if you look to the picture on the slide here, that is a sequence of DNA, and it has a microsatellite. This microsatellite is AGG, and it's repeating 11 times. So this is actually one of the microsatellites that were considered for this study. It's a little simplified, but it's the basics there. So what is PCR? This is another important component for the project. PCR stands for polymerase chain reaction. And there are different components in PCR. The DNA template, which is the DNA that contains the target sequence you're looking for. DNA polymerase, which is a heat-resistant enzyme that generates new DNA sequences, new DNA strands that are complementary to the DNA template sequence. And then with DNA polymerase, there's Taq DNA, which is most commonly used. But there's also top DNA polymerase, and that's what this research uses. In PCR, you also have primers. Primers are short segments of DNA. They're between 18 and 23 base pairs, and they are complementary to the target sequence you're looking for. They help to set the frame of the DNA so that DNA polymerase doesn't just copy the entire genome a bunch of times. So that way you have just the segment of DNA that you're looking for. And then last but not least, there are nucleotides. Those are DNTPs, A's, T's, C's, and G's that are used to build the target DNA strand. So these are the building blocks that DNA polymerase copies in so that the DNA can be amplified. From there, you make more DNA, and after 20 to 35 cycles, the sequence is amplified billions of times. And here we can see a thermocycler that is used for PCR, and it's the brand that's in the lab. So this is what we've been using. Methods. So this is what we're actually doing for the project. We've gone to different field sites, we've done sampling, and we have a list of the cultivars that are there. We've done chemical processing, and we're in analysis at the moment. DNA has been extracted from them, and we're doing PCR. The first step is the field sites. So we had growers that partnered with FSU to obtain those hemp samples. In 2019, there were 6 growers, and those are points 1 through 6. And then in 2020, there were 3 growers that were partnered with FSU, and those were 4, 7, and 8. These field sites are located in Garrett County, Washington County, Frederick County, and Hartford County. So, hemp sampling and cultivars. Sampling was done when the hemp was harvestable. To collect the samples, 1 stalk from the top ring of buds was trimmed off per plant, and then the leaves and buds were collected and placed in separate 50 milliliter centrifuge tubes. Those samples were then stored in coolers with wet ice until they could be placed in the negative 20 degrees Celsius freezer. Can I just give me a second? Okay. Just a sec. All right, so there were different cultivars in the study. There are 29 different cultivars that are here, and they are listed alphabetically. 22 are listed. There are 5 anonymous varieties, and then there are 2 that the grower didn't know what he was growing, so those are mystery samples 1 and 2. There's a lot of different samples here. My personal favorite is probably Charlotte's Gift, but there's a lot of fun names. The plants over here on the picture are stray cat samples that were at the field. Next is chemical processing and analysis. For chemical analysis, all the samples were lyophilized. The lyophilized samples were run until 20 to 35% of the original mass remained. The 2019 samples were sent for testing at Morgan State University, which we partnered with for the study. And there, those samples are analyzed for cannabinoid and terpene content. The 2020 samples are ready to be sent for testing. All right. DNA extraction. DNA has been extracted for all of the samples. We use the DNeasy plant mini kit from Kyogen, and the DNA concentration of the samples we used ranged from 2.7 nanograms per microliter to 123.9 nanograms per microliter. There were multiple replicates made per cultivar, and the low concentrations were dropped. So anything lower than 2.7 was dropped from the study. There was a final of 92 DNA samples that were diluted to 3.0 nanograms per microliter for PCR. So now, PCR. To do PCR, you need primers. So we used some validated primers and selected them from previous studies based on their ability to accurately distinguish species of hemp from one another and their reliability when used. So these are from various studies, and some of them are actually from a police STR multiplex kit for drug determination. We have 10 different primers here that we could potentially use depending on how well they work. There's also novel primers. These novel primers were generated. So overall, firstly, I needed to analyze the genome for potential microsatellites that could be there. And those generated over 1,000 microsatellite options. From there, we ran it through Primer Blast to determine primer composition, and Primer Blast selected different primers that worked, and those were only for some sequences but not others, so it further narrowed the search. We selected for self-complementarity and off-target binding, so if any of the primers bound to themselves or bound to something else besides where we wanted them to, those primers were out of the study. And then we selected the primers that remained based on similar melt temperature, primer size, and percent GC. So melt temperature is the temperature that you run the PCR at. So we need it to be the same, so that way we can make sure that the primer works well. Primer size should be similar, so that way you don't have one that's really big and the other really small. And then percent GC is just the percent of guanine and cytosine that are in the DNA. From there, it narrowed down the microsatellites to about 50. And the best of those microsatellites were chosen and are listed here. So those are the top 15 microsatellites that were analyzed. The chromosome number that they're from, motif, size and base pairs, melt temperature, and product size are listed. PCR is being completed on all the samples. For that, we used AccuPower Hot Start PCR premix. In every PCR reaction tube, in addition to the premix, there was 15 nanograms of DNA and 5 microliters, 2 microliters of forward and reverse primer as the baseline. It was varied depending on how well the primer did at different concentrations, and then water for a total volume of 20 microliters. For PCR conditions, we had 5 minutes at 94 degrees Celsius, and then 35 cycles of 30 seconds at 94, 40 seconds at 50, and a minute at 72. And then there was a final elongation of 72 degrees for 5 minutes. Gel selection and primer verification. So we had to choose what sort of gel to use for the PCR samples. Initially, there was a 2% gel used, but it had poor band visualization. And because we need that band visualization for the project, we switched to a 3% for a better resolution. So you can easily see on this picture here that on the 2% gel, you can barely see the samples, and on the 3% gel, you can clearly see 2 different bands. Those are the same PCR samples, but they just are run on different gels. This PCR verified that the primers CAN 100110 and CAN 2913 worked. So all of the validated primers that we used have been verified. Now, results. We used a Rubisco gel to verify that DNA was there. We're starting to get the results to be statistically analyzed. And to analyze things, we need to make sure that all of the DNA samples have amplifiable cannabis DNA. We used Primer Blast to generate Rubisco primers, and then we ran the PCR. And we found that all of the DNA samples have amplifiable DNA. And this top band on each of the rows is the desired product. So, yay, all amplifiable DNA. Anux 201 is the first primer that we tested. It is completed. The DNA samples show different alleles for this microsatellite. So if you look on the gel here, you'll see that some have 2 bands and some have 1 band. If there's two bands, they're heterozygous. If there's one band, they're homozygous. So on the top row, you can clearly see there's some samples with two bands there. Those are heterozygous. And then there's a run of samples about halfway through the second row, and those are all homozygous. This gel will be analyzed to determine the relative size of each band to the gene ruler 100 base pair ladder that was used. So those are the ladders on either side of the gel and in the middle. So what's next for the project? We're going to continue to run PCR and gels on each of the primers. We're going to include at least 6 validated primers and 6 novel primers. We're going to convert the gel images into data sets based on the band size and run analyses to determine genetic relatedness determination. So we're going to obtain the chemical data, run the analyses on that, and determine the chemical composition too. And then we're going to compare the genetic relatedness and the chemical composition and find correlations between the two things. Any questions? Any questions, anybody?
Excuse me?
Mhm.
Could you tell me a little bit more about the top DNA polymerase, like how it differs from the TAQ and why isn't it used as often?
Oh, so the difference between the two types of polymerases, it depends on the organism that it was isolated from. So Taq DNA polymerase was isolated from a bacteria in hot springs. It was the first polymerase that was isolated. And thus, for a while, it was the only one that could be used. So there's been a lot of studies to verify that. Top DNA polymerase is a newer contender that is it's outperforming Taq DNA polymerase at this point. Any other questions?
Yeah, I have a question. So you were talking about the bands in your PCR testing and how they were hetero in the other one. And could you define those?
So heterozygous means there's 2 alleles. So it's a diploid organism. So that means there's 2 copies of each chromosome. And each chromosome can have its own size of its allele. So on a heterozygous, there's 2 different alleles present. So 2 different products that can be found. And if it's homozygous, it has the same allele, same product.
Okay, thank you. And I think there's some questions in the chat for you.
Okay. I'm going to see if I can access the chat. There we go. There we go. So what can hemp be used for? So hemp can be used for like a bunch of different things. Hemp fiber can be used for textiles or rope or hempcrete, which is actually like a type of concrete made from hemp. And then the seed can be used for, we can actually eat the seed, or we can use it for oil, or it can be animal feed. The flower can be smoked, or it could be processed into essential oils like CBD or THC. For the other question, why do people only harvest the female flowers? Female flowers they have a higher concentration of CBD and they have more trichomes. The male flowers really don't. But the male flowers are good for things like pollination and for making seed hemp, because you need both genders to make the seed. And hemp male flowers, male plants actually have been used for doing fiber as well. It's considered that they have finer textile strength and stuff than the females. How hard is it to tell the difference between a male and a female? When they're early in development, it's really hard. But when it gets to the point where they start flowering, It is kind of easy because the male flowers have bell-shaped yellow flowers, and the female flowers have this really bunched structure, which I think I've got pictures of it on the slide. Well, I mean, this is part of one. So it's a very bunched structure that has female flowers just everywhere. Any other questions?
Yeah, hi, Janae. This was really, really good. Thank you so much. I enjoyed your research. I have a quick question. Actually, I have two questions. Number one, when you plant hemp, is that like any other plant? Like, would you plant that in the spring, or like, would you? And when would you harvest it? Like corn, or what's it compared to that I would be familiar with?
So it's a summer plant,
Okay.
planted sometime in June or late May, and it can be harvested.
No, I was just coughing. Sorry.
All good. It can be harvested in September to November. It
Okay.
needs short days to mature the flowers, so it needs a lot of dark at night and only a little bit of light during the day, compared to, normal crop plants like corn, which need a lot of light.
And another question I have, like 25 years ago, I was in Holland, and everything you're talking about, from the oils to clothing to rope to you name it, there were so many things being made of hemp over there. And, of course, it's 25 years later, and it seems like we're kind of getting to that point. Do you have any thoughts on that? I just think it's kind of funny when I think it was just so common and it wasn't an issue at all 25 years ago. And now it's like, now you go to the store, you can buy hemp oil, and you can go to the vitamin store and you can get all kinds of things that are medicinal, and clothing. You can buy, towels, ropes, shirts. And I just was wondering, what your thoughts were on that.
I think that, well, the era that it was really made hard to grow was around the time of prohibitions being made on many different drug substances.
Yeah.
And that's part of the reason why it was affected so badly, because there was just a lot of concerns about people using it for the wrong reasons. And then by the time that there was also a lot of synthetic fibers being made. So hemp wasn't competing as well with other things with more developed structure. So when it came time for the Controlled Substance Act, it had pretty much stopped being grown in the US. And that kind of just solidified its place as only drug. But at least now we're figuring out that there are other uses for this plant that are really helpful to society.
Perfect. Thank you.
Now we have less than a minute left on the chat, so I'm just going to stop sharing now. Thank you all for coming, and I hope you enjoyed the presentation.